development space, and how you've got examples globally of how this is happening, and currently the context and the continent where we have a lot of offshoring. So I want to ask my colleagues now in the South African space, what would it mean to you if we have a big data infrastructure in South Africa set up with population database? How does that help you work better?
Yeah, obviously we have the ClinVar is one of those databases. There's many others that we can use as geneticists to see if someone else has found a variant that we identified. But what happened in Africa with our wide diversity, we would have many variants that has never been described. So what happens is now you sit with a novel variant and you don't know what it means. So this database would help us to see, do you find these variants more in patient context, or is it in the normal population? And we recently had, just if I can use an example, I'm trying always to keep a bit, not to give examples because I can get totally carried away, but we had a very interesting TP53 variant recently that we identified in the beta isoform of the variant. So actually my first PhD student, it was Dr. Armand Peters, like a few years ago, he found it. And we reported it as a positive result. So immediately we got these questions, how can you be sure about it? But I mean, this patient, after genetic counseling, it became clear they has got the Lieframani syndrome, so the phenotype starts to fit. The patient came to us with a request from the oncologist, Marita, something must be going on here. She had 3 times new primary cancers. That fits in with TP53. She had radiation and chemotherapy. This is not something you should give someone with a TP53 mutation. So just to bring back the fact that although we want the population databases with specific disease cohorts where we can compare, the clinical information and having that background is crucial. So just what we did now is, because we had such an enrichment of data from one family, we then obtained ethics approval to write a case study. But this case study has now sort of going forward, and at the moment I have a PhD student. I've got no clue about, in vitro or ex vivo studies, but I could find someone at main campus, Prof. Annemart Engelbrecht, and she has got a wonderful student and she's busy to do ex vivo studies. It was always also a very big story of what type of functional studies will we do? If we do this, what answer will we get? How will it fit in? And I just came back from the PathRed conference, and on the bus, Professor Schneider, was previously my head at anatomical pathology, and I told him about this case. And I said to him, can you maybe do the immunohistochemistry, look at the TP53, staining it? And he said, yes, if you give it to me, I'll give you an answer tomorrow. So another co-author going on the paper that probably already have 20, but the point is, if we can see the example of one case at a time and what we learn from that and put that back into the database. So the database isn't only about screening 100,000. It is about that one that you found and then follow on. And for us to be in the environment where you can make use of research funds. Otherwise, it would have died just there, and it was just sitting there, nobody would follow through.
Thank you so much. I'm going to hand over to Janine to also answer.
Yeah, I'm going to try and be quick because my mind was just going, which example do you want me to use? So the Lancet recently published a paper based on, I think it was Lancet, the PREPARE study, which is a genomic test covering pharmacogenes. And they showed a 30% improvement in using that over 5 years in 7 European countries in preventing adverse drug reactions. 1 in 12 people will be admitted to hospital in South Africa because of adverse drug reactions. That test includes lacks genes that are critical in, wait for it, metabolizing TB drugs. Can you imagine the implication of taking a test like that and imparting it? Look, it's still going to make a big difference, but we need the background information because they are missing, genetic variants that just don't exist in narrow genetic populations. So I hope that conveys.
No, that's excellent. I want to open the floor for questions before we can close the session.
Thank you so much for a wonderful session. This is great to get a lot of feedback from the private sector and how it's working in that environment. But how do we bring in the government into supporting? I think Gretchen mentioned some of this earlier, where it works in other countries, like in Dubai we have G42, where in that environment the government has supported the genomics facility to get a lot of that information. How do we start something like that in Africa, South Africa, to get this entire genomics agenda going forward? Anybody that wants to answer it can answer that.
Facilitating.
Yes.
I need to answer that question. Okay, so I think there is this appetite in our government to develop a scaled population genomics program, and that's what we're trying to do. Obviously, we are a resource constrained country. We're not first world. We don't have a huge budget, and so we look at developing funding partnerships. And I think my colleague here as well, my fellow government organization, TIA, introduce Mohotlo. Come stand here, Mohotlo. No, because we're going to join hands, and we actively pursuing developing this national program and a national database for because we will never be able to enable personalized medicine or precision medicine without this. And we've heard incredibly well from our current research base that you don't have enough data. And we see examples globally of how countries actually support this. And the reason why countries need to support it, and essentially it's because, otherwise you have all of this data offshoring and there's no access for it for our current research base. And we're never going to be able to build an ecosystem in healthcare if we can't invest in genomics. And that's where we're at. We need to use cutting edge research to do that. So any other questions?
Thank you for a very exciting session, and some of the suggestions that you make or that we listen to are very interesting for South Africa. So talking about precision medicine, my question is when Janine, I think, spoke about TB drugs, we always consider adults, we do not consider children. And my problem is that when you look at TB especially, and you look at children, the adverse outcomes from administering those drugs in kids is even worse than the TB itself. So what is the sort of, angle, or what would be the proposal or suggestion that we can take regarding children?
Thanks.
I'm going to get into so much trouble. Last year, Professor Linda Gale Becker was on a panel with me in the main hall, and so I speak to her expertise in something that shocked me. We know that clinical trials have a dearth of females, and we know that they don't include kids. And so my quick answer to you, and she advocated it so strongly, it was almost a thought of putting children in a clinical trial. But if we don't put them in, we don't know what the values are, right? We don't know what the numbers are. I don't know how I can make a stem cell the equivalent of a child versus an adult. So from my area of expertise, I can't, but her advocacy for this was so strong, she convinced me in 30 seconds. And for me, that's just a quick answer. I hope that helps.
Thanks. Kelly Duplessis from Rare Diseases South Africa, and really, really insightful stuff coming through. But I just want to ask the question in terms of what investment is being made in terms of making this data and the terminology, the processes, the projects, etc., relatable to the patients. Because as a patient advocate, I sit here and it is very, very high level, and I see the lack of that. I see the lack of genetic counseling on the ground. I see the lack of patients actually understanding what contribution they can make to lay research and actually getting involved in these sorts of projects. And if we don't have patient consensus, well, we're not really going to be able to get it over the ground. So what sort of investments being made in that regard?
I don't know whether,
I can speak from other countries' perspectives and maybe throw the question back to our sort of more government-related colleagues here. For South Africa, from an international perspective, in terms of partnerships that we're involved with, specific funding around consumer and patient education is part of the program, as is also planning upfront, more so for the most recent programs over the last couple of years versus the sort of Genomics England at the beginning, in terms of health economics and the actual delivery. Something that is systematically in short supply and also not well funded enough yet when we get to execution, though, is in the genetic counselling side of things. So up front, the other thing that we've seen take place to help inform and request funding to be able to afford to include education as part of this is to conduct consumer or community surveys up front. So to really get an understanding of both within the individual consumer, I guess, patient level, what's their understanding of genomics or multiomics? Have they done any gene testing? Do they believe it's going to make a difference? So that's the other thing as well. And so we've seen a lot more when we've done those surveys up front. It can provide really compelling evidence choice points for governments to then choose to fund that as part of the incoming program, knowing that at the end you're going to need it to be able to translate that into the clinic. I guess in terms of what's happening within South Africa in particular, I'm not sure if that is part of the mainstream program.
okay, from a South African program perspective, I think at the moment we've just focused on the development of the scale databasing and the infrastructure and the actual sequencing and so from a clinical perspective, I think that we need to build that in, like you suggest, like other programs have, and we will take that suggestion definitely. And I want Marita to answer from from a current perspective where she actually advises patients and what's the experience.
Yeah, so this new app that I refer to, that is used to basically do a wellness survey for patients. It started very small. We have a paid genetic counselor dedicated to the project. We currently have a clinician. She did a PhD at Stellenbosch. It's a wonderful clinician. She just loves to be involved in this multidisciplinary approach. So what we do is the P4 medicine, it includes as one of the P's participatory. So for me, one of my PhD students actually went on to do a genetic counseling course, and I supported her all the way because I realized how important that was. And if you do not have a genetic counselor that's actually trained a bit outside of the mainstream genetics so that we can bring all the aspects together, the pharmacogenetics, the lifestyle trigger genetics, and the diagnostics for where the genetic counseling is now done mostly in familial context. The counselors that work with me, they're branching out into the other areas. And I can say that the latest one that worked in my team, she initially said to me, but it's not my place to talk about those things. And now she had a poster at our academic year day about wellness and how that brings it to the patient on the floor. But you need a counselor that can put everything in perspective. And one of her conclusions was that by explaining about not just you don't have a high penetrance gene, we cannot exclude that you maybe have one, but we cannot do anything. By actually taking it back towards these other there's three aspects. It is, the familial risk, the lifestyle trigger risk, and the therapy-induced or associated risk. And if a genetic counselor can be trained to look at all those aspects, just think of the benefit, but it can't be just anybody. And then we have the clinician that look at the medication that the patients use. And it's amazing to have her now in the team because she immediately knows what is the generic names or vice versa of the medications that we don't know. So yeah, and it's a long COVID project, which the MRC also funds.
Thank you. Any more questions?
Thank you so much for a very informative session. I want to follow up on the idea of multiomics being the future. Where are we locally, globally, and where to? Thank you.
So this is a multi-pronged answer. So I think we start locally, then globally.
Okay. I mean, I'll do it in 1 minute, but we need an hour. Um, I'm aware of other groups who have started looking into this. In a paper published last year, in 2022, of the, I think it was 200 to 300 papers that had published multi-omics data, 2 came from Africa. That should give you an indication of where the issue lies, but also gives you an indication of, with the right infrastructure and funding, where the research is going. So certainly from commercial points of view. Things that we're looking at now, and we discussed this earlier on, it's really expensive to do that. So we're working with data sample sets, clinically curated data sample sets in the tens, to do genomics, transcriptomics, proteomics, and we're just picking 3. Well, we're doing multiple genomics, but that's another thing, ChIP-seq and all kinds of epigenetics. But so we're looking at that, and we're trying to balance that. We also have to balance it with the disease that you're looking at. So, for example, liver disease, cancer, maternal child health with regards to hypertension. We're looking for high impact disease cohorts. So we have to be careful. But for some of them, we can do 10 samples. We've got access to 20,000. But we don't have the funding for that, so we have to start. That's the local situation insofar as I know, and I'm happy for anyone else to add. Do you want to add global? Yeah.
As Janine said, her and I were talking about this earlier. So from a global perspective, layering on obviously transcriptomics, single cell, methylation, proteomics, and the ability to do that is the existing larger programs that have taken place. Now we're systematically adding proteomics data to that. When you look at Our Future Health and you look at UK Biobank, Singapore is doing the same. One of the challenges, as Janine was saying, it's cost and differential infrastructure. And so something for us as a company that's been sort of right on our radar since we began was almost this infinity loop of applications of omics, and how can we continue to build them out, leveraging the same, I guess, hardware architecture from genomics that can expand to other applications, so that you're not having to pay 5 times the cost of a single sample to get all that multi-omics. You need to get all of that basically in what the one cost is. So, it's very much where our technology roadmap is going, and it informs the decisions that we make in terms of technology and applications, because that really is, obviously, where you start to get the really valuable insights.
Can I add one quick? We keep referring to cost, and it keeps coming up, and perhaps I haven't alluded to some of the ways we're approaching this. The UK Biobank consortium partnered with Regeneron and GSK, if I'm not mistaken, and they created a model where those companies would pay for all the sequencing, do all the sequencing, send it back, but I think they had a 9-month moratorium in being able to work on it, and then it was publicly available. Now, as researchers, initially we were a little bit concerned by that. Is that something we should do? But it takes 2 years to publish the paper anyway. So I'm fine with the company paying for all of that. So we're changing that kind of, as long as we make sure it's coming together, it's a method of getting to the end. So those kinds of moratorium models are things that we are exploring responsibly and ethically.
Thank you so much. Can we take one last question, and then we're going to close?
Hi, everyone. I think it's a very beautiful idea, of course, the idea of the genetic counselor supporting your PhD student in regards to multidisciplinary research. Because a lot of research now, especially quality research, relies on people collaborating with each other. So I just feel like funders should do more to support the multidisciplinary approach, because you get a student, realizes that they need more knowledge in order for them to achieve what they have to do. and they have to learn a new concept from scratch, and then it becomes quite a tedious process whereby whereas if they were collaborating with another student, another master's student, or another PhD student in a field, in the particular field of interest, it would take maybe 2 years instead of it taking 4 years when they're alone and having to learn a new concept. I feel like it becomes a situation whereby our funders, people who fund the students from NRF or what have you, need to realize this, or the supervisors need to realize these multidisciplinary research researches that have to be done, and identify that they have to do funding that's collaborated. For example, as a chemist, for example, we'd have do something whereby we get, let's say, funding from the government or what have you, that supports a dual research thing between a chemist and a geneticist, or a physicist and a mathematician. And it really needs to start from up top, whereby they realize that in order for us to make more quality research, we have to collaborate kids so that they get the bigger picture and learn how to collaborate from a young age, from the master's honors even, if possible, so that growing as this generation of new researchers, we don't just see things from one side. I don't just see things from a chemist. I see things also from the perspective of a mathematician, what have you, and we work together. Because the thing is, it becomes a situation whereby, yes, she comes to your genetic counselor, but he's busy and he has a lot of projects going on, and he's gonna take time nurturing certain skills or certain things that she wants to do, because he still has a whole list of things to do. So whereas if I'm doing it with another peer, things go faster, and yes.
Many of my PhD students are actually medical doctors. I love it to have them as students, because then I would always link a lab scientist as far as possible to that clinician. So the clinician would do their research, recruit the patients. We have a research assistant to help with the informed consent. That was actually the one person that went on to do a genetic counseling course later on, just beside the point. And then I had another student that sort of zoomed in on one aspect of her study, where we found something that was not explored because it wasn't included in the PhD study. And there's so many of these aspects that comes out along the way, and then to have another student working on that. And that is actually our latest publication that was published. The title also says, genetic counseling, lifestyle adjustments, and change of medication, because the clinician could bring in a pharmacogenomic study to look at aromatase inhibitors that increase your risk for osteoporosis. But then some people already had osteoporosis at baseline, and if we didn't do the study at Tygerberg Hospital, those people would have got the medication without some protection or either change them to another medication, tamoxifen. And then most of those patients ended up to have very low vitamin D levels, which is an undisputed risk factor for osteoporosis. So this new student from the lab came in, and he looked at the vitamin D receptor genes. And we could see that for the baseline cases, that the vitamin D wasn't enough to explain the difference, why some people had osteoporosis at baseline and others not. But then by adding on SNPs from the vitamin D receptor, he could get significant difference. So this article was the best example of what I call pathology-supported genetic testing. It was postmenopausal hormone receptor positive patients. If we have worked as in a whole group, we may not have picked up the changes. And by the fact that we did whole exome sequencing, those people were already screened out at Tygerberg Hospital for having founder mutations, for BRCA1 and 2 or extended testing in a panel. But once we did WES, we also had ethics approval to obviously look at the breast cancer genes, and we found still 10% of those whole exome sequences that had another mutation that was pathogenic. So it was just this wonderful study. And then the best of all was that at Tygerberg Hospital, I then asked the genetic counselor to counsel the patients. She drew blood again, and then it went into our workflow at Bloemfontein, where they routinely do the NGS for panel testing, including BRCA1 and 2, and confirmed the mutations that we found in a research environment, and that research results could be fed back. So this is a new, like a new model that we developed. Do research, look wide, but bring then certain aspects back into the diagnostic platform. I hope it's clear. It's just so beautiful.
Thank you so much. I think we've had such a wonderful discussion today with so many recommendations. I don't know if my panelists want to have any last remarks. Otherwise, I want to move to close the session and appreciate all of you for attending. And if you have any further questions, you can please accost these panelists after the session. Thank you very much.
So my last remark is that we need to have increased access to these tests. So what would we do? You start with something that is well accepted across the world, BRCA1 and 2. We put those variants that are common in our population into a point of care assay. It would help. It is supposed to be done by a genetic counselor with assistance if needed, so that they can see, can I stop when I found this variant? And if I feel unsure about point of care, because we do not have SAPRA approval yet, just repeat that one of the 9 variants, and we have examples of 2 patients that had surgical appointments already, and the results could come out in time for that surgery to decide on a double mastectomy instead of maybe not. So that was a very important thing. And that same assay is also for PARP inhibitors. It's now become very important. So it is for pancreatic cancer, ovarian cancer, breast cancer, and prostate. So the access and the plan with that point of care is to take it into rural areas, just as a step, just open the gate. And then it would go to other people in South Africa. Many of our labs offer that to go on with next generation sequencing in negative cases. And immediate action, even if you just have to retest, a small, less than 1,000 rand test. Access is my thing.
An API plant for malarias, anti-malarials in Nigeria. We also have Msizi Pharma, but Eugene will speak on that. There's also CPT Pharma, who we have been working with over the past, I think it's now 2 years. We've given them 2 grants to work on dolutegravir and molnupiravir. So before we go to APIs, I think the key message here is that local manufacturing is actually not a new topic. As Africans, we have been manufacturing since the 1930s, and there are about 650 manufacturing plants across 29 countries in Africa. So I'm going to specifically talk about the ARV value chain and about the ARV APIs and the work that we're doing in the ARV space. And for me, personally, ARV and HIV was a very foreign idea. It was a problem that my parents suffered from. But we need to remind ourselves that we still have the highest burden of HIV in Southern Africa and the highest burden of people living on antiretroviral therapies. I know for us, the younger generation, it's now my life, my choices. But HIV is still real and is still a problem. So that presents a market opportunity as well as a public health opportunity. So a group of funders, or partners as I like to call them, met this year. The meeting was organized by the South African Medical Research Council in Johannesburg. And we started to discuss how can we really tackle this project or problem of API manufacturing in Africa. So we selected two lighthouse projects, one looking at injectable ARVs and another one looking at diluted gravier. And this is essentially the group of partners who are working together to solve this problem. So what we decided to do was to develop a business case or feasibility study for diluted gravier API as well as injectable ARVs. And I'm just going to give some highlights on the injectable ARV study. So just a few highlights from what we have learned so far. So what we know about ARVs is that the SADC region manufactures only 15% of their own ARVs and 24% of the other drugs, which is relatively low. And that there is no API manufacturing. We are importing almost everything or everything from India and China, and they give us a discount of about 15 to 10%. What I found interesting was that in last year's Global Fund tender, 153 tons of diluted gravier landed on the shores of Africa, and about 66% of that actually came into Southern Africa. So what that shows is that there's a very huge market for diluted gravier on the continent. We are the biggest consumers. So I'm going to now look at South Africa as a specific case study. Why South Africa as the only country in the SADC region that really has a public tender for ARVs. Most of the countries have quite a small public market and a very large donor market. So this gives us a very big opportunity to start thinking about market shaping interventions and government-led interventions. My apologies for the flashing. Yeah. So what I think the key message here basically is that the South African latest tender is valued at 11 billion rands. Of that 11 billion, it's a value of 9.5 billion dosages of ARVs, and 58% of that are DTG-based products. So what I'm trying to show here is that there's a very large market already available. I'm trying to build a business case for this. And then what we're seeing is that 55% of the last tender was actually awarded to local producers. So what we're doing, we can literally easily close the value chain by ourselves. So this is just some details showing how much diluted gravier actually lands into South Africa. On an annual basis, we're looking at 91 metric tons of diluted gravier actually landing into Africa. So I won't speak much about the processes, but what I want to highlight here is that there are many numerous processes to manufacture APIs. I'll leave the chemistry to the experts on the panel, but what is just to demonstrate is that of the four that I've shown, two of them, the NMU one, Paul Watts will speak about it. It's a South African process available here, and the medicines for all process, chemical process technologies, pharma, CPT pharma, have already localized to this process. So what this shows is that there is technological maturity for us to be able to manufacture locally diluted gravier. We tried to look at the cost, the raw material cost, to see if it was quite profitable. And what our research kind of shows here is that at least for the Aurobindo process, it's patented, and it has a lot of reagents that are used, so it can be quite expensive. But for the medicines for all, even at a low yield process, which is the process that CPT Pharma have adopted, for example, they're quite cost competitive. That's basically the message I'm just trying to share here. So I won't bore you with the Excel sheets, but after punching around a lot of Excel sheets and a lot of numbers, we got to that the design capacity for an API plant, looking at the current market share in South Africa, as well as by Global Fund and other partners, you could have a plant of about 150 tons per annum, and you would be relatively profitable, provided that we are considering final dosage formulation for TLD, single entity DTG, and pediatric DTG formulation. So in conclusion, basically, the South African market presents an opportunity for us to essentially do some form of market shaping work, both supply side and demand side, for us to have a profitable API plant in Africa. There is an opportunity for an African API manufacturer to actually enter the market. You would need about 500 million rands, which is about $30 million of capex for initial investment. But we really now need to work on the offtake agreements, do a lot of demand side support, as well as some supply side incentives. In one of the previous slides, if you notice, the cost of financing is quite expensive. So we really need to work together as a government, as governments and development agencies to provide some innovative financing solutions for us to make this project a success. And basically, this is the end of the story that I have. Thank you very much.
Right, so morning everyone. My name's Paul Watts from Nelson Mandela University, and I always like this talk titled Drugs for Africa, Let's Go with the Flow. So I'm pleased to say that my data, I think, very much agrees with what Faith has just been talking about in her talk. Living in Port Elizabeth, if you chat to somebody in the street, they will tell you that Aspen, which is literally 15 kilometers from my office, is a pharmaceutical company. Of course, as we all know, that's not the case, and what all of the companies do in South Africa, the formulation companies, is buy the white powders, the active pharmaceutical ingredients from China or India, and at the moment, our part in the project, or the South African part in the project, is actually making the tablets. Now, the value of South African pharmaceutical imports in 2021 was about $3 billion US. So my proposal today, which I think correlates with what Faith was just talking about, is let's just use that money differently. One of the speakers in the previous session was saying that South Africa is resource limited. That